So welcome to the monthly JETZON open call. So I'm very pleased to say that we have Alexey Gol this month, who's going to be talking to us about Arctic squid. So without further ado, Alexey, over to you. Thank you.
Hi all, thank you for having me here. And today, indeed, we discuss the Arctic squid. Okay, so today we discuss Arctic squid. It's life and specifically life history reconstruction using carbon and nitrogen stable isotopes, and which implications for functional biodiversity does it potentially have. I now work in GeoMar, it's in Kiel, as Marie Curie postdoc fellow, and it was recently a paper which is a part of this fellowship. It's a result of our international team. I'm grateful to my collaborators and colleagues who participated in this. They all are listed here. So today we discuss, it's a brief summary. At first, as a concept for functional biodiversity, as one species often can have several roles, and what can we do about that in terms of like modeling and ecological reconstructions? How do we access ecological roles, specifically using stable isotopes? Which implications this correct ecological periodization can have to our example species, and which implication correct ecological periodization can have to the whole ecosystem? So let's start. At first, it's Barents Sea trophic web. This picture is from Institute of Marine Research. Maybe some of you know it. It's Barents Sea ecosystem. So phytoplankton, zooplankton, larger zooplankton, squid and fish, and benthos, and also demersal fish. And then there are seabirds and marine mammals. And let me draw your attention here, for example, like small pelagic fish, let's say herring, and once it hatches its larva, then it grows, it's small, then medium, large, and it consecutively changes not only its size, but sometimes its habitat, and also its prey spectra and its predators. So different size, different diet, different predators, and it results in several, in one species, and even one individual, if that one individual is lucky, occupies several ecological roles during its ontogenesis. So from functional point of view, in functional biodiversity, it can be functioning as several species, not as one. So let's discuss one particular example. Our example today is no fish at all. It's Arctic squid, Gonatus fabricii. It's the only squid species in the Arctic, and its common name, vernacular name, is Boreatlantic armhook squid, actually. But if we look at this range, you see it's mostly in the Arctic. What is so curious about this species? So except for its vast range, only in Nordic Basin, Norwegian and Greenlandic seas, it has biomass for more than 20 million tonnes, and from that, 1.5 million tonnes consumed by sperm whales alone. So it's very important for top predators such as marine mammals and seabirds. But also, as it hatches, it's only size like 3.5 millimeters in mental length, and this is body length. It's used as size measure in cephalopods, mental length. So once it hatches 3.5 millimeters, so in this size, of course, it's eaten by crustaceans and then by fish. And as it grows, it becomes also prey for fish and for top predators such as sperm whale. Also, it's a carnivorous species, so in turn, it also eats many different Arctic inhabitants. So it's important as both prey and predator. Also, within this vast range, it reproduces only in some particular areas, and then it must have vertical migrations. And also as it grows, it has so-called ontogenetic descent. So larvae and small squids, they're epipelagic, and as they grow, they go downwards to meso and then to bathypelagic layers. And that's why it became relevant for deep sea studies. And they reproduce always in bathyal layers. And after reproduction, females, they undergo the jelly transformation, stop feeding, and live as bionts for the eggs, which they carry in this position as the drawing here shows. So this is a quite interesting species. And if we apply stable isotopes, we can decipher its life history. And talking of stable isotopes, it's a small methodological things. If I assume not everyone might be working with stable isotopes, and it's worth to discuss it a little bit. So the most applicable stable isotopes in ecology are carbon, C13, and nitrogen, N15. So if we speak of carbon, the regular carbon, C12, is like almost all carbon in the world, almost 99% is the regular carbon, but heavier stable isotope, it's carbon 13, that's the one we use in ecology. Also, there is a carbon 14. This is used in radioisotope analysis with half-life about 6,000 years, but we're not discussing this one today. And in nitrogen, the regular nitrogen, everything consists of is more than 99.5% is nitrogen 14, but nitrogen 15, that's our target stable isotope for ecological reconstruction. So when some animal eat something, heavier isotope atoms, they preferably stay in the body, and lighter regular atoms, they are preferably excretable. And this is the basis of this ecological application. So as stable isotopes are preferential to stay, so with time and with some specific circumstances, they will accumulate, and that's where ecology came into play. Carbon can be used to track habitat, where as a living, and not directly, but it's more of what was the primary source of carbon in the food web they come from. And nitrogen can be used more directly using specific equations to estimate trophic level. For this equation, so we have values of our target species, and to estimate its trophic level, we need a baseline from which we estimate its trophic level. So the base here is an assumption that each next trophic level, there is like a trophic enrichment factor, so-called step between trophic levels, by which nitrogen stable isotope accumulates and increases. And then the baseline regarding which we estimate this. As a baseline in the Arctic, we usually use herbivorous copepods, Calanus. The herbivores, so trophic level 2, phytoplankton trophic level 1, Calanus trophic level 2, and we apply to this equation. And trophic enrichment factor, there are several approaches. Which one can we take? So the classical one, 3.4 per mil. And there is Arctic specific one, 3.8, and this is exactly the one we use in this study. But there are ideas like a scaled approach, so it is changing between different trophic levels. So, and we can see cephalopod beaks here. In their mouths, they have beaks, which function as jaws to eat their prey. So why do we use beaks for stable isotope analysis, but not muscle? So at first, cephalopods are very agile, so they easily can escape from our gear when we try to catch them. But large fish and seabirds and marine mammals, they actually function as biological samplers. And in scat analysis of seabirds and in stomach content analysis of large fish, as exemplified by this Antarctic toothfish, for example, we can find cephalopod remains. And soft body parts are rather fast to be digested, but beaks stay there for quite a time. So beaks are in a way easy in this relation. And then just imagine if cephalopod is caught, and then it keeps somewhere in collection, and it is being fixed in either, usually they are fixed in ethanol and formalin. And in muscle and soft body parts, we cannot use it for stable isotope analysis because isotopic composition and soft body part is changed. And bigs are better again. So in big stable isotope composition does not change due to fixation. So even long time fixed bigs, they can still be used for stable isotope analysis. And the last, but not the least, in fact, it's very important here, bigs can function as archive tissue. So they are hitinos, and once they are formed, they reflect isotope composition which came from the diet in that specific moment, and it is not changed after that point. So, and as cephalopod individual grow, newer part of the beak reflects its diet in the farther parts of ontogenesis, but previous they just remain as it is. So beak function as archive tissue. So if we pick up from this assumption and we know that growth direction is from anterior to posterior parts, what can we potentially do here? So if we powder and mix the whole beak, it will be a proxy of whole life. And if individual was small, medium, or large, then what small squid could potentially have been eaten or large, it will all be reflected as a proxy of its whole life once the beak is powdered and mixed. And as I told you, we know the growth direction. So if we cut the beak in halves, and then we take respective pieces from the most anterior to the most posterior part, and then we can have a trajectory from within individual, from first to last point. What has it been growing? Where has it been more or less living during all its life? So we apply two different approaches, both to beaks in such a case. So we had more than 100 beaks of different sizes to assess the population. And if we have 4 large beaks to assess individuals, each one from 11 to 17 subsections. And also, as you can see here, why subsections are of different sizes? Because we know the most important and actually majority of changes are in the beginning of the ontogenesis. So we need a higher resolution. And we do not lose much if in the later part of ontogenesis we have like a more coarse resolution. So we apply these two methods, and then we have both population and individual assessment. What else is often done in stable isotope analysis is analysis of isotopic niches. So from a simple B-plots to more complicated Bayesian approaches. But these consecutive pieces, which arise from the single beak, they actually do not fall under assumption of sample independence. And the most commonly used package, which is called Cyber, is not applicable in this case. So we use another one, which is called Nishover, and this one is better to use in Swanson. You can read more about this package. Also, isotopic niche is not really equal to trophic niche because carbon, as we mentioned, it also reflects the habitat. All these results were, at first it's the samples. The samples are from East and West Greenland and from the Barents Sea for population analysis, and samples for individual analysis, they all originate from West Greenland. These results are already published, accessible by these links. And now let's have a look at our results. So carbon and trophic level. We use trophic level using the same equation I described just recently as a proxy of nitrogen to as application for nitrogen values. So both isotopes, and in this case, of course, trophic levels, they show significant ontogenetic increase on both individual and population levels. Carbon values within single individual, it can be increased up for 2.2 per mil, and this is very, very big increase. It's not usually like that. And it suggests these changes are beyond what diet can explain. So they also change their habitat, not only their diet. And in terms of trophic level, it's up to almost 2.5 trophic levels within single individuals. Again, this is a very large increase. It's not really usually like that. And it suggests very strong changes occur in species ontogenesis and even within one individual ontogenesis in terms of their diet. If we apply isotopic niches, so these are subsections from 1 to 14, and subsections 1 and 2, when the squid is small, they are very different. And as the squid grows larger, from subsections actually 5 to 14, they are very similar to each other. So if we try to, in a way, combine what both isotopes say about this increase, so yes, there is increase, but then after some point, squids just tend to live more or less in the same place and eat more or less the same thing. And from 31 to almost 190 millimeters, they more or less reach their adult isotopic niche. So how did we get these sizes from these subsections? So if we measure some certain amount of beaks, which came from individuals, not from stomach content, but from the field, and we also measure individual itself, then we can have an equation. And that is how we can back calculate then the size of individual which it potentially had when the beak was of the size of the subsections. Then we back calculate this, and then we know also the smallest squids of these particular sizes are the most different, and as they grow, they all live in the same place and more or less eat the same food. If we try to access, okay, what exactly are they eating then? From stomach content, we know it's crustacea, and within crustacea, it's largely copepods, FOZs, and shrimps. And other than crustacea, these are arrowworms, hetagnata, cephalopods, and fish. And also, Gonatus fabricii is kind of the only squid species in the Arctic. So cephalopods is largely arises from cannibalism. And as we can see, it's rather high proportion. So it's also a highly cannibalistic species. So then we already know when changes occur in carbon and nitrogen values, trophic levels, and isotopic niches. So based on that, we pick up which subsections we should compare in terms of diet modeling. And that's why we pick up these 4 groups from all groups we have. And what can we see here? In smaller squids, subsections 1 to 5, so below 35 millimeter squid size, crustaceans constitute more than a half of their diet. And at that time, fish and squid, they form like complementary patterns. They contribute together, fish and squid, less than half of the diet then. And once the squid is above 47 millimeter, it's the opposite. Crustaceans contribute less than a half of the diet, and combined fish and cephalopods, they contribute more than a half of the diet. So somewhere we can see in between, in the sizes, they change their prey spectra from more crustaceans to more fish and cephalopods. And if we have a more detailed look to crustaceans, within crustaceans, the smaller squid, they have more or less equal proportions of copepods, FOZs, and shrimps. But all other groups of squid are significantly different. And the largest crustaceans of these 3 groups, shrimps, they dominate the diet of all but the smallest studied squid groups. If we try to derive some implications, at first let's combine this and have a periodization of ontogenetics of our target species in terms of both diet and habitat. So below 20 millimeters, they are purely epipelagic and largely prey on copepods. Then 20 to 50 millimeters epi to mesopelagic. And given all what we already discussed, that's exactly where the switches in their diet come. So at first, they switch within crustaceans from copepods to FOZiacea, FOZiids, and shrimps, and then they switch in a more global sense from crustaceans to fish and cephalopods. And after 50 millimeters size, they already live in meso and bathypelagic layers and largely prey on cephalopods and fishes. So the trophic level continued to increase and they continue to go deeper and deeper, but largely they already reach their adult diet proportions over 5 millimeter size, 50 millimeter size. And actually, as we remember, the largest recorded can be up to 40 centimeters. So they just go deeper and prey on larger fish and larger cephalopods, but proportion is already as it is here. And only females, they stop feeding after 20 millimeter, 200 millimeter, 20 centimeter body size. And after reproduction, they have this jelly transformation and just guard their eggs in this position. Potential pitfall here, what we should do in the future is paralarva from 3.5 to 7 millimeter body length. What do they eat? But of course, they would be epipelagic. We already know that. And most likely they will be eating copepods. So if we add paralarva, we potentially can have even better resolution of this ecological periodization. If we try to apply this in a more global sense, so first application for cephalopod ecology in general, and let's link it to the Arctic ecosystems. So many ecological shifts early in ontogenesis and also very high trophic level, almost up to 5.5. It also is known for Antarctic cephalopods, but not in temperate or tropic waters. We can say right now, like cephalopods on the poles indeed have the higher trophic levels than they do in temperate and tropical ecosystems, because now we need more studies from non-polar areas of other oceans, Atlantic and Pacific, where trophic level of cephalopods has been identified correctly. And what do we mean here, like correctly identified trophic level? Let me just briefly show you this thing. So global distribution of stable isotope baseline and carbon and nitrogen. So carbon have like, okay, relatively well, not really well-known patterns, but at least there are some patterns in carbon distribution. So for example, we can see decreases from equatorial to polar, right? Also, we can see decreases from neritic to oceanic ecosystems. And also we know it increases from pelagic to more benthic organisms within the same ecosystem. And variation is more or less about 16 per mil worldwide. But in nitrogen, there is like literally no obvious pattern. And variation is much higher than carbon, it's more than 20 per mil. And what happens if we take global samples and just compare raw values of nitrogen? Let me just exemplify it here. So this is one of our past studies with Henkian and other colleagues. It's about deep sea cephalopod known as vampire squid. On this map, we can see global samples from Pacific, from Atlantic, almost reaching the Arctic, it's almost in Iceland, and also from Indian Ocean. And on this graph, black dots are the nitrogen values. I would say from 6 to 16, right? And they show increase. But the real pattern is the white dots. This has trophic level. So once we apply correct baseline, and this nitrogen, raw nitrogen values started to have ecological sense, what can we see? Trophic level worldwide is actually from 3 to 4.3, I would say. So variation is much lower than in raw nitrogen values. And what is the most important, actually trophic levels are decreasing during ontogenesis, not increasing. And if we compare raw values from different ecosystems, the real pattern will remain hidden. So that's what I mean here. We need more studies from different oceans where trophic levels are identified correctly, and then we can say, is it like indeed trophic levels of cephalopods are high in polar areas, or it's just because the trophic level now it's better identified in Arctic and Antarctic than outside polar areas. So once more studies will appear, we can say, is it indeed true about the poles? So who are, let's say, colleagues of our Arctic squid on high trophic level from 5 to 6 in Arctic ecosystems? So we remember that Gonatus fabricii is 5.5. Who is also here? It's of course marine mammals, specifically the top predators, also seabirds, and especially those of them who have partly avian diet themselves, seabirds who eat other seabirds, also large piscivorous and large scavenging fishes, also there is Greenland shark, and also there are large scavenging benthic invertebrates who eat carcasses on the seafloor. And also our Arctic squid is here. Why? What are potential reasons that squid in the Arctic have so high trophic level? So squids are able to eat prey which is much larger than themselves. And we couple this with high degree of piscivory and high degree of cannibalism, and exactly that's what it results in. Large squid, very cannibalistic and eating large fish, they obtain this high trophic level in polar ecosystem. Another Arctic ecosystem applications might be the ecological role of some species shifts several times in their entogenesis. So here we can go beyond cephalopods. It can as well happen in fish and crustaceans. And so in functional biodiversity, one species can be represented several times. So in terms of functional species, one species can be represented as several because it has several ecological functions. If we combine ecological role in our example species Arctic squid, so for example, these ones are all from West Greenland and from small, medium, large. If we combine it, we can see it has the largest niche from among studied so far Arctic invertebrates. This example is also from West Greenland, and shrimps, crabs, scallops, copypods, whatever, they all have not that wide isotopic niches, despite they also have some size differences. So species who change their ecological role several times, of course, they have very wide isotopic niches, unlike those who do not change their ecological role so actively. And now we also have correct data for future ecosystem modeling, at least for our example species. Which conclusions can we potentially arrive here? So first example, Arctic squid illustrates the importance of assessing different functional roles consecutively occupied by a single species. Again, it's not only cephalopods, but also happens in fishes and crustaceans. So both population-based and individual-based stabilized top analysis, they can be jointly applied. And in fact, I would say they are recommended to be jointly applied for ecological roles assessment, because if we apply both population and individual-based assessments, it gives us a better picture and better resolution. At least 100 species for population base, and if we split them roughly in small, medium, and large, and especially if we're using like some hard body parts, archive tissues from small, medium, and large, then we just powder them and use this as a proxy of a life of individual. And then we have like 10 or 15 more, the largest, and then we cut them in pieces and have individual trajectories. And then we unite the results obtained by different methods. And then here we go, that's the basis for ecological role assessment. And again, data presented here, it's a necessary baseline in cephalopod biology and Arctic ecosystem studies. For the modeling, we can take exact sizes of squid when they live in this particular area and eat this particular food. I want to acknowledge European Union on this grant agreement and also my co-authors. Thank you for attending.
Thank you very much, Alexey. Yeah, very interesting. So let me see if there's any questions in the chat. So if anybody would like to ask a question, please just raise your hand or type a question into the chat, or to tell the truth, and just open your microphone and ask a question. So while people are thinking on questions, so what is it that I'm just trying to sort of understand that the squid has this ontogenetic descent, and so that it sort of moves downwards with age. So what is the advantage of doing that in terms of, what is it that has led the squid to have that pattern of behavior? Because, naively, you might think that having the really small stage in the surface would make them more susceptible to predation. So I'm just curious to know what it is that's potentially influenced that ontogenetic descent.
Yeah, I think there is a trade-off, because as they grow, they become bigger, and then, for example, seabirds can target them and eat them even more effectively than before.
Right. So
the trade-off, I would say, is as follows. So they are abundant, and they go for copypods, which are ubiquitously abundant. And despite squids are being eaten as well, they eat copypods and grow, and they from copypods they switch to larger crustaceans. But as squids themselves became larger and more easily targeted, they go already deeper. And when the squids became really big, they descend even further to hide from visual predators. And then in the depths, they already go for fish and for their own species, as we remember they are cannibalistic.
Okay, yeah, that's interesting. So in terms of the reproduction, so did you say that's a bathypelagic phase, so that sort of deep water? So are we talking sort of below what, sort of 2,000 meters for that stage?
Yeah, the reproduction, the large individuals, which can potentially be mature, or in late maturing stage, they usually encountered below 500, but the reproductive sites, it should be at least below 1,000. And along the Norwegian shore, there are 2 sites which are deeper than 2,000 meters.
Wow, interesting. And then the larvae return to the surface then, so they sort of migrate back to the surface.
Yeah, it was only once when the females with eggs were actually captured. It was one paper from 1997. Right. And then there are several papers when they record paralarva on epipelagic layers. But how did they get there? Right. The guess could be that but it was never observed, per se, it was never observed.
Okay. Yeah, fascinating. Thank you. So are there any questions for Alexey? Say if you want to add anything in the chat, or just open your microphone and ask anything, then please do feel free. I mean, I've got another question if nobody else has got one. Sorry. Interesting talk, so I've got lots of questions. So you said that Arctic squid have got very wide niches. So once again, what is it do you think that's influenced that? I mean, is it a case of, well, I'm just curious to know what you think might have influenced that, because, I recognize that other species are very specific in terms of their niche. So I'm just curious to know why you think that might be such a wide niche for Arctic squid.
Yes, of current state of knowledge, it's also in Arctic and also in the Antarctic, they reach very high trophic level. And for Arctic ones, we also know they have the widest niche. So once we will have a more definite answer, if temperate water squids have actually lower, or do they also have higher, but we are just not aware of that now. So maybe what can be the case, that's because of a reduced fish diversity in the polar areas.
Right.
That would be my guess, but
So,
yeah.
Okay, so you're saying that the squid sort of expanding into that opportunity.
Yeah, because like in the Antarctic, cephalopods are very diverse, more so than in the Arctic. But fish fauna, fish diversity is it's less so in the polar areas than in tropical.
Interesting.
So maybe this is the case.
Is there any major difference in the temperature effects on physiology between cephalopods on fish?
Yeah, it also can have something to do with that. I never worked with physiology, so I cannot say it outright, but
yeah, very interesting, very interesting. Last call for any questions or comments for Alexey. I say if you prefer, then I'm sure Alexey won't mind if you want to email in later if you do have
sure.
questions. And I say, please do check out his paper. And I say, Alexey, if you can send me the link for the paper, then I'll embed it in the talk when we post it on YouTube so people can take a look.
Yeah, I will definitely.
Excellent. Right. Then if there aren't any more questions, I'd just like to thank Alexey once again. Thank you all for joining the call. And please do keep an eye open for upcoming calls. There'll be one in April, towards the end of April. And do also take a look at previous talks on the JETZON YouTube page. But otherwise, thank you all very much for attending. And thank you, Alexey, once again for an excellent talk.
Thank you. Thank you.